Nicole, if you want to go ahead. It is a pleasure for me to introduce today's speaker, Dr. Lucas Westermann. He is in internal medicine at the University of Friedberg. He's also a clinician there and a senior research fellow. And he's going to tell us today about some heterogeneity that happens in clonal cell lines that may contribute to our variability in phenotypes that we see. So I think a very interesting topic. Lucas, I'm going to turn it over to you. And again, thank you very much for taking the time to meet with the group. Look forward to hearing what you're saying.
Thanks for the kind introduction. Let me just share my screen with you. So now you should see my slides. Is it correct?
Correct. Looks good.
Okay, perfect. Yeah. Thanks, first of all, to Terry and Owen for inviting me to today's RRC Scientific Roundtable. The title of my talk is Wild-Type Heterogeneity Contributes to Clonal Variability in Genome-Edited Cells. And in fact, I'm really thrilled to have the opportunity today to share some lessons we learned the hard way with the ADPKD scientific community today. So please allow me at the beginning a very brief introduction to ADPKD. As you know, ADPKD is characterized by the emergence of fluid-filled cysts that are derived from a very specific cell type, the renal tubular epithelium. And we know that each of the cells in the renal tubular epithelium harbors a germline mutation for, in most cases, PKD1 or PKD2. And we know quite well that the cyst-initiating event is a somatic mutation or loss of heterozygosity that gives rise to cyst formation. And we know quite a lot about the gene products of PKD1 and PKD2. We know that PC1 and TRP2 act in concert and form a receptor ion channel complex. And we know quite well about the localization of these proteins. They are in the membrane and the cilium of the renal tubular epithelial cells. And although we have learned quite a lot about these proteins, there are still some fundamental questions that need to be answered. We do not know what is the activator of this complex, and we do not know what is the signal that is transduced by these two proteins into the cell. So after having studied these proteins for many years, we have to ask, why do we lack deeper insight into PKD function? And the obvious and perhaps true answer would be that it's really complex and complicated, but perhaps there's also an additional aspect to consider. Most of what we learned in the last years was derived from studying animal models, or to be more precise, to study murine models. So by genetic manipulation, we analyzed the consequences of this manipulation and learned well about modifier of polycystic phenotypes. And although we have learned quite a lot, there are still some limitations in studying animal models. All of you know that it is very time-consuming business. The degree in which we can get new insights into molecular mechanisms is limited. And I think, which is also a very valid point, is that the animals are not amenable for high throughput screening and novel drug discovery. So with this in mind, I think it's valid to say that there is an unmet need for a robust cell-based ADPKD model. And in recent years, many researchers of this community have addressed this need by trying to establish cell models, such from the lab of Benjamin Friedman, for instance, Raphael Krammann, Owen Woodward, and many more, just to name a few. And in the Köttgen lab, we also were dedicated to generate a cell-based ADPKD model. At this point of my talk, I want to emphasize that this talk is not about a novel discovery in ADPKD, but this is more a methodological approach or more a focus on a phenomenon that perhaps many people have observed when studying genome-edited cells, the phenomenon of clonal variability. And this is what the talk is about, clonal variability and the underlying causes that can be huge confounder in experiments involving genome-edited cells. So let me briefly explain. A few years ago, when novel genome editing techniques such as CRISPR-Cas9 and TALENs emerged, then we, of course, wanted to perform knockouts of the disease-relevant genes. And Of course, you can take several cell lines for studying PKD function. And in our case, we decided to take IMCD3 cells and MDCK cells, and from our perspective, for good reasons. They are differentiated, so they have apical-basal polarity, they harbor cilia, they are from the disease-relevant nephron segment, easy to propagate, and I think we can consider them as rather stable. For deleting the PKDs, we use CRISPR-Cas9, and in this case, PKD1. And I think it's really relevant for the next slides to highlight our genome editing strategy. We really wanted to delete the whole coding sequence, and that is exactly what we did and what we validated to really be sure that in all experiments involving the PKD1 knockout cells, that we do not have hypomorphic alleles or residual protein function. When we established our PKD1 knockout cell line in IMCD3 cells, of course, we started to do experiments. And one of the pathways in which we were interested was the HEPO signaling pathway. You know that one of the effector proteins is called YAP. And when we studied YAP function in our PKD1 knockout, there was an observation. We saw that there was a downregulation of YAP compared to wild-type cell lysates. And this is odd because perhaps as you remember that there was a very interesting paper, I think in 2080, where Dr. Pan and his colleagues showed that there is a connection between HEPO signaling and ADPKD. And what they basically found was that they identified the HEPO signaling effectors YAP and TATS being modifiers of polycystic kidney severity in murine models. And what they also found was that there is a drastic upregulation of YAP levels in PKD1 deficient kidneys. So this, of course, was in contrast to what we observed with this PKD1 knockout clone. So when we tested a second knockout clone, then we saw the opposite. We saw a drastic upregulation of YAP. And when we repeated this experiment, it was quite consistent. So after having done 3 rounds of this experiment, there was a significant difference between PKD1 knockout clone 1 and PKD1 knockout clone 2. The explanation when you have an observation like this would be that, of course, you mix something up and one of both clones is no PKD1 knockout. And of course, we validated again and we saw, okay, there's really a lack of the whole coding sequence. We can be sure that these are true knockouts. So in this case, what many researchers refer to are off-target effects. So are off-targets at play here? Let's have a look. We know that CRISPR-Cas9 enables precise gene manipulation on target. So this is exactly what we intend to do. We want to delete a certain gene. So here indicated on the left side of the picture in blue, you see the CRISPR-Cas9 complex. In green, the guide RNA that directs the CRISPR complex to a genomic strand, which shows homology between the guide RNA and the genomic strand. And if there is homology, then next to a so-called PAM, a protospace adjacent motif, double strand breaks can be induced. So really important to know is that the prerequisite for inducing double strand breaks relies on the homology between your guide RNA and your on-target strands. And just when you have this homology, you induce a DNA double strand break. This DNA double strand break is most often repaired by the cellular repair mechanisms by reconnecting the DNA strands. And this is called non-homology end joining. But this process is very error prone and often results in indels. But CRISPR-Cas9 can also manipulate the genome off target. And this is very important because when we design a guide RNA, then it should have selective homology. So meaning that there should be mismatches between your guide RNA and other parts of the genomic DNA to decrease the probability of off-target cleavage. So in this case, when there's no homology, there's no cutting event. With this in mind, we can say that clonal variability is not sufficiently explained by potential off-target effects for now in basically 3 reasons. when generating the two knockout cell lines, we use the same guide RNAs. Both of them, which we used, have low off-target prediction, and we also observed the phenomenon of clonal variability when we use tailends in the past, which have a very low off-target prediction probability. And all of this in the context that we used stable cell lines such as IMCD3s and MDCKs. So the question was, what is the reason for clonal variability that we observed in this experiment, but to be honest, also in other experiments and in other cell lines? Because we realized that there must be a big confounder in our experiments involving genome-edited cells, we had to step back a little and take a more global view how we generate our CRISPR-Cas knockout cell lines. And we did it, like many do, by clonal isolation. Just have a look at the workflow. So you start with a wild-type population of cells and then transfect your cells with a guide RNA and the CRISPR-Cas and the Cas9 complex, and then you perform a single-cell sorting. And with this single-cell sorting, just a few cells will have the desired genome editing event that you screen for by multi-well PCR. And this workflow results in the end, after validating your clones, in a monoclonal PKD1 knockout cell line that you compare to a wild-type cell line. But the prerequisite that you have a valid experiment is that your starting population of wild-type cells is homogeneous. So we asked, does the clonal variability that we observed result from differences in the starting wild-type cell population? And this question was addressed by generating monoclonal wild-type cell lines. So we took the parental wild-type population and just did a single-cell sorting. And this led us to have several cell lines which we arbitrarily labeled wild-type 1 to wild-type 5 as indicated here in different colors. And the obvious experiment that we did with these monoclonal wild-type cell lines was to test again YAP levels. And the surprising result was that there were big variance in the YAP levels when considering and comparing our different clones. And when we repeated these experiments, it was again quite consistent. There were quite relevant differences in our monoclonal wild-type cells. So of course, now you can argue that hyposignaling YAP is also dependent on culture conditions. Perhaps there is another variable driving these observations. So we tested other ADPKD related signaling pathways. And what we found was that quite consistently that there were differences in levels for some proteins such as PPERK or CAM kinase delta or PAMPK. And when we again repeated these experiments, we found significant differences between the clones. So how about other readouts? Is it just related to Western blots? Let's check. So in the introduction, I mentioned that it would be highly desirable to have a cell-based model in which we could screen compounds for novel drug discovery. So we tested susceptibility of our cells for several cytotoxic agents. Here in this example, you see that we tested the viability of different monoclonal wild-type cells into polyclonal wild-type cell line and over several doses. So we performed dose response curves for viability. And when we tested cisplatin, which is quite a cytotoxic agent, then we found there were no real differences in viability between the clones. But when we tested a more specific inhibitor, it was quite a striking result. In this case, we tested methotrexate, which is a dihydrofolate reductase inhibitor, and we saw that there were huge differences between the clones. Here indicated in red, you see, for instance, wild-type 2, which has only a modest decrease in viability compared to a drastic decrease in viability in green, wild-type 5. And it's, of course, quite obvious when we quantified the data, there were significant differences over all doses and quite remarkable over 3 biological replicates. So Then we asked if these differences in monoclonal wild-type cells can also be interrogated in a more biologically complex assay. And in the Köttgen lab, we try to establish a cell-based model where we try to induce IMCD3 cells to become tubule cells. So basically what you see in the lower row here of the image. And what we have in our assay are predominantly two structures, spheroids and tubules. And by genomic manipulation with compounds, etc., we want to see if there is a difference in tubule ratio. So what we have in this assay is here an overview of just wild-type cells. And I think what you can acknowledge is that we can induce these cells after 7 days to become quite nice tubules in most cases. And when we delete PKD1, then we see an impaired tubulogenesis. And this is exactly what we try to interrogate with this assay. And for quantification, we use a neural network that we have trained to recognize structures in these images and to classify also in an unbiased fashion structures into blue tubules or red spheroids. And then we can automatically analyze and quantify the data to see if there is a difference in tubulogenesis. And so we checked our 5 monoclonal wild-type cell lines, and again, there were quite a big differences between these clones. You can perhaps not acknowledge it in the small pictures, so I magnify, for instance, wild-type 2, where we have really nice tubules, and now compare it to wild-type 3, where we have an impaired tubulogenesis, and this is really quite a striking result if you consider that this is something that we have observed in the PKD1 knockout experiment. And when we quantified the data again, there were this was something that was, of course, statistically significant, for instance, between wild-type 2 and wild-type 3. So what we learned from these experiments was that wild-type cells have a phenotypic spectrum. And this has implications for the way we generate our knockout cell lines. So if you start with a cell line, a parental cell line, that is in fact not homogeneous, but polyclonal, then your workflow will result in having a monoclonal knockout cell line that you compare to a polyclonal wild-type cell line. And at this point, you can ask, why is this important? Valid question. Let's have a look. What we did is we did a sham CRISPR experiment. So we did exactly with the old workflow that I showed you to generate knockout clones. We took out of a pool of these polyclonal wild-type cells, we isolated a single monoclonal cell line, and then without any genomic manipulation, we isolated RNA and did an RNA sequencing. So when we did this RNA sequencing, we did a differential gene expression analysis of just wild-type cells. And I think this is the most striking result that we had on this journey. You see by just comparing wild-type cells that there were hundreds of differentially expressed genes despite adhering very stringent cutoff criteria. And that was really a striking result. Out of fun, we did a gene ontology analysis, and it was really interesting to see that just by comparing wild-type cells, the most enriched terms were receptor regulator activity, signaling receptor activator activity, and receptor ligand activity. All terms that you could easily misinterpret in an experiment involving PKD1 knockout cell lines. But in fact, they are just the result by comparing wild-type cells. In addition, we learned something else by these experiments, by these transcriptomic comparisons. We did a principal component analysis, and what this means is basically that principal component analysis reduces complexity of a very large data set, such is the case for transcriptomics. And what you can see here is in red, the technical replicates that cluster quite well, indicated here in red on the right side, in contrast to the polyclonal wild-type cells. And what this experiment already indicates is that the transcriptomes of monoclonal wild-type cells are more similar than those of polyclonal wild-type cells. So with all what we learned here, there must be an updated way to generate CRISPR-Cas knockout cell lines. So what we did is, in the first place, or what we have to do is to start with a homogeneous population of monoclonal wild-type cells. So first, what we had to do is a single cell sorting. And if you do this workflow, in the end, you will have a monoclonal knockout cell line that you compare to its isogenic monoclonal wild-type control. And this is, in fact, exactly what we did. And I have to underline that this was quite a journey, because you have to see that we already had a lot of knockout clones in our lab, but with the knowledge that there is a huge confounder in these knockout cells, we basically had to start all over again and generate all knockout cells again for the relevant genes we were interested in. So on the next slides, I will show you data about or comparisons of the knockouts that we generated with this new protocol, and we term them monoclonal knockouts in comparison to those knockout clones that we generated with the old protocol. We term them polyclonal knockouts. From the beginning of my talk, you remember the YAP experiment where we had contrasting data in YAP levels between the polyclonal knockouts. And when we now did this experiment again with the newly generated monoclonal knockouts, we saw that there were no differences between these monoclonal PKD1 knockouts. And also some other interesting observations when we use polyclonal PKD1 knockout cells to investigate AMPK levels. There was a downregulation in these clones. When we had monoclonal knockouts, there was no difference at all. And this was also true for PMPK. So just a few examples. So these Western blot experiments also indicated that monoclonal knockout cells seem to show less variability compared to polyclonal knockouts. But we wanted really to be sure if this is the case. So we asked, does the generation of isogenic wild-type cells really reduce clonal variability? And for that reason, we again made the experiment with the transcriptomics. Just a few minutes ago, I showed you the sham CRISPR experiment with hundreds of differentially expressed genes by comparing a monoclonal wild-type cell line to a polyclonal wild-type cells. So now, how about the same experiment with a clone derived from a monoclonal wild-type cell line? So just to illustrate it, we started with a homogeneous monoclonal wild-type population, isolated a clone, which we termed subclonal wild-type, and then again, without any genomic manipulation, we isolated RNA and did again RNA sequencing. This is the result. Having done the same experiment just with the homogeneous starting population, we just have a few of differentially expressed genes. And in fact, this was so striking that we repeated this experiment with several other clones, but we always got more or less the same result. So when we quantified this is the differentially expressed genes which were interrogated with the old protocol. We saw that there were 800 genes differentially regulated. When we applied the new protocol and did the transcriptomic analysis, only a few differentially expressed genes. And I think it is valid to say from this experiment that the novel genome editing workflow that we used drastically reduced false positive phenotypes. So to illustrate our findings, we learned that wild-type cells have a phenotypic spectrum as indicated here by different colors, and by transfecting this heterogeneous starting population with the guide RNA and the Cas9, this workflow will result in having a random monoclonal wild-type cell that becomes in the end a successful knockout. But we learned that the monoclonal wild-type cells are derived from some kind of phenotypic spectrum. And in fact, when you use this workflow, in the end, perhaps what you see is only wild-type variation and has nothing to do with any genomic manipulation. So to get rid of this wild type variation in your experiment, we would recommend to start with a monoclonal wild type cell line and then isolate this wild type cell line, transfect it, and then in the end you have a knockout cell line with an isogenic control, and you eliminate the confounder of wild type variation, and this gives you a much more better understanding in the analysis of genotype-phenotype relationships. With this, I would like to summarize my talk. We learned that heterogeneity of wild type cells is a major contributor to knockout variability. Very important to note that assumed biological effects can be driven by heterogeneity of wild type cells instead of reflecting a consequence of genomic manipulation. And we showed a novel genome editing protocol that drastically reduces false positive phenotypes. And also, I would like to give 3 remarks. Still, functional analysis of different monoclonal clones from our perspective is required. We know that rescue experiments ensure the identification of biological effects. And we have to acknowledge that also inducible knockdown or knockout cell lines provide an isogenic control. If you're interested in more details, then please have a look at our publication recently published at Scientific Reports. At the end of my talk, I would like to acknowledge all the people who have done tremendously well on this paper, or the whole Köttgen lab, and put an emphasis on someone in our group which had the idea of systematically analyzing clonal variability. This was, of course, our PI, Michael Köttgen, highlighted here with an arrow. Otherwise, you wouldn't recognize him among the students and postdocs. Thank you very much.
Lucas, thank you very much for a scary and insightful talk. There are some questions in the chat, so I'm going to ask you guys to unmute yourself and ask your questions, if you don't mind. Rick, Con?
Yeah, that was a great talk. And as I said in my comment, I hope every trainee, student, postdoc, and PI sees this talk and the work and takes it to heart. I think there are a lot of really important lessons in there. But it also emphasizes the importance of what we call rescue experiments. Anytime you see a phenotype coming from knockout, if putting the gene back in does not reverse the phenotype, then that's a problem, which I think you didn't really address. But the other question I had is, and I think another comment is asking essentially the same question, kind of where did your first wild type cells come from? That is, you know, if you get a line from ATCs or if you generate the new, it should be clonal. But if it's grown multiple generations, it's going to diverge over time.
Yeah. So to address your first comment, I would really agree that rescue experiments are should be done in all your efforts in trying to investigate some consequences of genomic manipulation. Sometimes it's quite hard. For instance, with the PKD1, it's not that easy to bring back the cDNA. Perhaps for instance, in our experiment, this is something that we accomplished, but still, you can argue that proteins such as polycystin-1 need a narrow control of expression levels, which is not that easy to achieve in transducing cell lines with, for instance, CMV promoters. And the second question was more. Can you just give me just a single word for the second one?
So the source of your polyclonal?
Source, yeah. So ATCC, yeah. And I would agree that perhaps of course, after cultivating your cells for weeks or even months, that of course can lead to genetic drift and can alter your wild type cells. But in this case, we used ATCCs in quite a low passage. Yeah, and this was really for us striking to see that IMCD3 cells should be monoclonal, or they have monoclonal origin. And it was really disturbing to see for us that they are that heterogeneous. Yeah. But still, perhaps a remark, these kind of experiment that we did at the end of this talk when we had a subclone derived from a monoclonal clone. In fact, we incubated these cells for some weeks, like 6 weeks. And I think we have to acknowledge that still the variability after 6 weeks is quite low. Yeah, so there is certainly genetic drift in these cells, but not as much as we initially thought. But still, what I think which is really important when having generated knockout cells derived from a monoclonal cell line, that you really have to cryoconserve a lot of vials of your cells, and especially of your isogenic control, that you never run out of your control cell line.
Agree. Kenneth and then Reem.
Thanks. Very nice talk. So it sounds like your study suggests there's a lot of heterogeneity among the polyclonal wild type population. So how do you know you end up choosing a monoclonal cell line that you knock out, let's say PKD1? How do you know that it's representative of the overall population? And then more importantly, the in vivo situation, often, and these cell lines I presume are immortalized, so there may be some effects of immortalization as well that make it less representative of what we expect to see in the animal.
Yeah, great question. This is, of course, something that we debated a lot in the beginning of this project. I think you really have to be sure what kind of monoclonal cell line you start with. For instance, when we had the isolation of different monoclonal cell lines, we saw that there was, for instance, one cell that wasn't able to generate cilia. And of course, this is not a cell line that you want to start with investigating a ciliopathy. Yeah. So in our case, we wanted to have it as representative as it might get, that it has levels of polycystin 1, detectable normal growth behavior, cilia, apicobasal polarity, everything that you can check. But of course, in the end, you don't know if it's really representative. But I think which is even more important to study genomic manipulation was that you have in real and isogenic control. And if we have to choose between these two possibilities, having a more polyclonal cell line, and which perhaps represents or is more representative, but then of course, after having all these results here, I would always go for the monoclonal starting cell line.
Reem?
Lucas, thank you very much. I'm Reem Mustafa. I'm actually an outcome researcher, so I don't do any basic research. And I have to admit, one of the struggles I always have when I listen to some of the, basic science research that is presented is all the methods questions I have in mind. So I want to applaud you for actually taking this critical look based on the methods, because I do think there's such an important question in general in basic science, but I also want to pose it to the PKD RRC, is how much time should we as a community spend on kind of looking back and asking some of these basic questions that actually at sometimes may explain some of the inconsistencies in the findings, some of the studies that we can't replicate, and so on. And I work with a group called the GRADE, GRADE working group, which now this method is really established worldwide to evaluate certainty in evidence for outcomes and guidelines. And I always think about all the intersection between what we think about with GRADE for these outcomes studies and what should be applied in basic science. So really more of a comment than a question. I think my question to you is I probably will reach out to see if we can apply some of these frameworks to the GRADE work you've done here, because I think it has a huge implications, and I hope more of the group would be interested in applying some of these basic questions, if you may, to the models that are being used, whether cell models or animal models or so on. But a question maybe to the PKD RRC in general, Brad and others, is that something that the consortium is interested to supporting and maybe giving some guidance to the larger community do this, don't do that. Here are some of the issues and so on. But thank you. Really great talk. Appreciate it.
I fully agree.
Thanks.
There's two questions in the chat, one of which I don't know who it comes from, but I'll read it out anyhow. It's, did you synchronize the cells before you introduced CRISPR to the IMCD cells? And then the next one, kind of follow up on it, how quickly would a wild type clone diverge once you had made a clone?
Okay, the first one I didn't get. Yeah.
Did you synchronize your cells before you introduced CRISPR to the IMCDs,
No,
before you made
no.
your mutants?
No, we didn't synchronize it. No.
Yeah.
And
then
the second one is, yeah, the second question, how fast they divert. And this is, I think, something that partially answered before, that this transcriptomic experiment in the end, where we had cultivated our cells for weeks, yeah, and the drift, the genetic drift that we saw, the difference in the transcriptomic data was really quite low. So I think it is, yeah, it's not a very big genetic drift, at least in these IMCD3 cells, and of course we cannot generalize.
Okay, so the question I had was, I'll throw one of mine in here. So we had been doing some stuff with IMCD3 cells as well. We do karyotypes on these guys, right? And we will see in one clone it has this karyotype, and the next clone it has that karyotype. Have you characterized any of these impacts with karyotypic genomic changes that might explain some of this?
Yeah, very good question. Yes, we did. Yeah, it was not that easy to get the karyotyping done in murine cells, but yes, this is something that we did. And because, of course, you can ask the question, why do you see these really big, striking differences between the clones? Is the karyotype, is it responsible, for instance, structural abnormalities in your cell line responsible for the outcome? And in fact, we studied quite well in the end, and we couldn't see that there is a correlation of karyotype and phenotype. So we investigated different monoclonal wild-type cells, the PKD1 knockout cells, monoclonal, polyclonal, but in the end, we couldn't see a correlation. No.
Lucas and Michael, I'm going to have a question for you guys. There's been several requests that came to me in the chat about wanting to show this to students and trainees, your talk. And I would love to be able to do that. So if you guys are okay with us doing that, please let me know, and we will would like to use this as a demonstration of how to do it correctly and some of the caveats and concerns you have with cell-based culture studies.
Yeah,
sure, go ahead.
Sure, yeah.
I appreciate it, guys. That's because I think this is truly a demonstration of, you know, how it needs to be done. Any other questions? We've got a couple minutes yet.
So Brad, if it's okay, how would one access or download the talk?
It will be up on the PKD RRC website, and if you, I will find out how we can actually download it, or you could probably access this directly from the website. It'll take us a couple days to get it up there.
Yeah, I think you can access. I wasn't sure if you could download it directly.
Yeah, that I don't know about. We will have to.
Okay.
Terry or somebody like that will.
That'd be great.
You will be able to access it, but you may not be able to download it. So it will be there. It'll take maybe a day or two before it's there, but you will be able to see it.
Yes.
And YouTube.
Yeah, if you need to get it downloaded, let me know. I will see how we can manage that.
Great. Appreciate it all.
The file's probably too large to easily.
It'll be large, yeah. You know, the way we always end these roundtables, you know, so the PKD RRC is meant to be a resource for generation and distribution of resources. And clearly you've shown an important aspect of cell-based resources that we need. So is there anything that the center could do that would be of importance that would advance your guys' research moving forward?
Yeah, so I think one fundamental aspect that you all already address is that you can connect people, that we talk about the hurdles and the roadblocks in our field of research. But besides that, of course, we would be happy to have Cysteine-1 antibody that is more reliable in detecting endogenous levels of PC1. Of course, this is something that I think many people would wish for. Yeah.
Okay. So we have several things in the pipeline for that, but that
Okay.
is noted. All right, any final questions, guys?
So does it make sense to make these cloned cells available? I worry a bit.
So I do know that most of the clones and things that we are generating within, or most of the lines that we're generating within the center are derived from clonal cell lines.
Oh, they are.
They are. If not, we sure as heck will be making sure they will be moving forward.
You will now.
Yeah, we will now.
Okay.
So Lucas, I want to again thank you for a wonderful talk. Again, this will be available up on the website shortly, and we appreciate your taking the time to meet with us. So thank you.
Thanks a lot.
See you guys.